So my name is Jeff Shu, and I'm going to be talking a little about what I do. I'm a geneticist, so I'll be talking about genes. Not hipster genes, silly, but the genes that make up biology. So I'm from the Cleveland area. I work at the Cleveland Clinic, and I'm associated with Case Western Reserve University, is where I got my degree in molecular medicine, but really just what I really got my degree in is in genetics. This is what the campus looks like. It's quite beautiful on a summer day. I suggest you all go visit sometime if you have the time. There's a beautiful art museum there. And part of NIH guidelines is that we list our financial disclosures. I don't have any. I don't have any patents. I don't have any equity in pharmaceutical companies. And what I work on is cardiovascular disease. It's actually the #1 cause of death in the United States still. Roughly 30% of all deaths that occur are due to CVD. And this is despite large gains in fighting cardiovascular disease. So deaths due to coronary heart disease, in the blue dots listed there, peaked at roughly 325 deaths per 100,000 within the 1970s. So on the y-axis is just the death rate, and on the x-axis is just the year. A somewhat similar story to stroke, which is a related disease to coronary heart disease. And what I've put there in the arrows are some of the drugs that have been developed over the years. The red one is the first year that statins became available, roughly in the mid-1980s. And so that was one of the biggest drugs that affected the disease rate. Of course, I'm not all suggesting most of this steep decline in death rate is due to drugs, but also programs such as smoking cessation. So in a way, cardiovascular disease is sort of a victim of its own success. We've essentially lowered the death rate 70% to the level almost to where cancer is at the moment, all cancers. And so we kind of have to be a little bit smarter in how we develop drugs and maybe target the people that use these drugs. And so one way to do that, and we know that genetics and genomics plays an extremely large role. Not only can we be able to target these drugs more effectively, but also we know that through twin studies that these diseases are actually quite heritable. Roughly 50 to 60% of the variance in disease outcomes is due to genetics. And so the central dogma of nature really is DNA, which is the heritable component that we receive from our parents, is transcribed to a molecule known as RNA, and this then is translated into proteins. And we can do an association test between the DNA and the phenotype, because that's quite easy to do. We can genotype the genome across all markers relatively easy, and we can measure phenotype just as easily. And so what is the file you get out of genotyping? It's really just a diff. You have locations in the genome where there's differences, and for instance, on let's say chromosome 1, position 200, and the diff is against it's an arbitrary set sequence that is based off of Craig Venture's and James Watson's genome, the first two people that were sequenced across the genome in the early 2000s. And so yeah, it's just basically a diff file, and listed at every marker. So each one of these locations is a region that is polymorphic, meaning there's multiple variants at that position. And so a lot of this data is actually publicly available, accessible to really anyone. So the 1,000 Genomes Project is a large project that tried to characterize a lot of the rich variation that we see in the human population. And all their data is public. dbSNP is the database for the actual markers, so every marker that's been seen across all sequencing projects in the world. And so that's becoming quite large. There's over 60 million variants found within the human genome across the population. And so again, all that data is publicly available. This is government funded, so it's paid for by you, and so you should be able to access it. There's a really cool Rails site actually that lets users submit their genomes when they're genotyped by a consumer company such as 23andMe or FTDNA, Family Tree DNA. And this is the picture here is just a platform used to genotype. Each one of these dots is a marker, and the fluorescence tells you which, basically, what color it is, what tells you what marker it is at that given position. And so, yeah, so people have uploaded their genomes as well as their own phenotypes to allow really anyone to do this science. And I'll give some examples of this later. Another project's the Personal Genomes Project, headed by George Church at Harvard, where they're doing a more organized effort for volunteers to submit their genomes as well as give their full health phenotypes so that researchers and amateurs can do the science, not have to rely on NIH-sponsored groups. So what I work with, this data is quite big, as I said. There's, on average, the genotyping platforms genotype 2 million markers, and really to do population genetics, you need hundreds of samples. It's not quite, big as in the data that you guys probably work with, but it doesn't fit in Excel, at least the old version of Excel where they had a memory limit. So I consider it big, even though it might not be. And the one thing is when you work with a dynamically typed languages is that you lose a bit in speed, but it really isn't everything. Or at least in my case, because the taxpayer is going to pay for it. But no, I'm joking. And so the advice there is, the advice there usually is to drop down to a lower level language, right? Drop down to C, where there's a bottleneck. But I really can't do that because personally I'm probably not capable of doing that. But thankfully, there are actually really quite good tools out there now for, at least for just-in-time compilation. And so, if you guys, as a scientist, your peers will look down at you pretty badly if you believe in occult things. But sometimes you have to make an exception, and I believe just-in-time compilers are that exception. There are quite a bit of sorcery involved there that I don't understand. But what I do understand is that they do speed up the code quite a bit. So I'm actually a Pythonista, and I just give some examples here. I know benchmarks and all, but you see that using Cython, which is a C compiler, I'm using pointers here. It's the same algorithm, only I'm statically typing and using pointers for arrays. And a just-in-time compiler known as Dogma that's put out by Continuum.io. So I get the same exact speed up with the same algorithm just by putting a function decorator on top of the function. So in terms of, and that's not all, right? Because now there's also tools available that are heavily written in C and wrap a lot of the C libraries. So one of them is SciRuby. It's a collection of gems that do a lot of functions, such as there's a visualization software, distribution, which is a statistical software for running the various statistical distributions, such as the normal curve, minimization, which is basically minimizing a function, and the big thing obviously is working with matrices. And so NMatrix is sort of the big tool. It's currently been worked on quite heavily. It wraps basically Atlas, IPAC, or OpenBlast, or whatever your linear algebra library that you prefer to use. And so, yeah, so those are the tools available. And why I think this is important is because the sequencing costs have dropped dramatically over the past couple of years. So it's actually, in the green line, those are the sequence. It's actually dropped quite a bit faster than Moore's Law in the past couple of years. So the first human genome project cost roughly $3 billion to complete in the late 90s and the early 2000s. But nowadays, we can sequence a genome for less than $10,000. And the goal eventually is to try to get that to $1,000. And it's going to happen in the next couple of years. So the flood of data that's going to be publicly available is going to be huge. Other tools to really analyze this data, and more specifically, are BioRuby. It's a gem that is actually more useful for classical molecular biology. But the big one is something called BioNGS, or next generation sequencing. So that next generation sequencing is the main tool that has allowed the drop in sequencing costs. And so what I've shown here in this console is just basically the output of what you get in this data. The sort of the format that's sort of been adopted is known as a BAM file, or a binary alignment mapping, and each one of these lines is a read. And what comes out of these sequencers are roughly 300 million of these reads. And so, so it's quite a bit of data, and you basically, since we have a sequence, and these are short fragments, we can align that sequence to the known reference human genome generated by the original sequencing project. And so in addition to some of the Ruby tools I listed, I actually have my own open source analysis tool it's called Genda. It's just genetics and pandas, which is a data frame library in Python, similar to what you get in R. And IPython Notebooks is actually a really good tool. If you ever use Mathematica to do analysis and share that analysis, this is very similar. It's just it's a front end for IPython. And what's nice about that is that you can use Ruby in it as well. Although, just if you want to pass data between, let's say, Ruby and Python from plotting. So I'll talk about genome-wide association studies. So now you have the genetics. You're trying to figure out basically a novel pathway that wasn't found before that can potentially be intervened with drugs. And so one way to do this one recent technique is known as a genome-wide association study, where unrelated samples are parsed basically into cases and controls. And using the data that I showed previously, you just basically do a regression. This could be done with binary cases, like such as cases and controls, or with a quantitative trait. And so what you get out of this is basically something like this. On the y-axis, you see a -10, log 10 of the p-value, which is just the indicator of how significant the association is. And so you see a locus right here, a couple markers here that is significantly associated to whatever trait this is. And this was done in Genda. And this is an actual published study, which we published recently, where multiple loci within the human genome affect this particular disease, atrial fibrillation. And so there's thousands and thousands of loci associated with hundreds of traits. And you can actually see all this data published in a government database known as genome.gov/GWAS studies. So there's a wealth of information there done on, right now, probably more than 4 million individual samples associated with hundreds of disease. And so, once you get this hit at a genomic loci, what does it do? One of the biggest criticisms of these studies is that they hit in regions of the genome that didn't have a protein coding gene, that didn't have, express a protein. And so the question there is, are these real? And I just want to address that real quickly in that there's two ways that a variant within DNA can affect phenotype directly. One is to affect the RNA sequence, so this will actually change the structure of the protein and potentially its function. The other is to actually affect the protein levels itself. And so again, with this newfound sequencing technique, we can also sequence the RNA as well. And what you get here is you see a histogram of the individual reads, and that's an indication of how highly expressed that gene is. So each one of these boxes here is a read that you get, again, from the sequencer. And you can quantify this not just at a particular gene, but across the whole genome. So again, a lot of this data is actually publicly available. It's available at the Gene Expression Omnibus site. It's another NCBI site. There's more than 1 million samples that you can look at. A lot of those aren't human samples, but they represent the same kind of data in a variety of species. And so here's just an example of an association of gene expression data with a genetic variant. On the x-axis is just the variant encoding, and on the y-axis is just the expression levels of a given gene. You see that having one variant leads to higher expression over the other variant, and the heterozygous intermediate individual that has one variant and the other variant is an intermediate phenotype. And so we see when we do this across the genome that there's hundreds of genes that are affected by some genetic variant within the genome. So this is a very pervasive way of controlling gene expression as well as expressing phenotype. And recently, a study published out of Stanford showed the same thing. Showed a lot of this is again that this is extremely pervasive, that gene expression is highly controlled by genetic variation. And so again, this eventually leads to differences in protein expression levels that can affect disease. And one tool that we use a lot is something called pathway analysis. Is now that we find these genetic associations, is there a particular pathway now that we can intervene in that and potentially affect disease outcome? And so this is just basically an enrichment analysis. You take one set of data, you take another set of data, and see if there's more overlap than what you expect by random chance. And this is a good indication of that, that pathway may be extremely functional in disease pathology. And so, obviously all this is a basic first step to identifying novel pathways, but it's an extremely important step. And the data is out there to really even tease this apart, where it's not really just restricted to scientists off in a closed lab, where maybe some type of analysis was overlooked. And it really, I think it's an important step, because if you identify these pathways, you can design basically something called siRNAs, which is basically antisense. It basically binds to the given gene and knock it down. So that's an extremely potential source of intervention that's actually quite easy to do. And there's a lot of actually drugs in the pipeline right now that do this, as well as identifying potential small molecules that could knock down or antagonize the protein as well. And so, so I'm actually mainly a Python guy. There's a lot of great tools out there for there. And again, as I said, the Science Ruby ecosystem is actually growing quite fast. And I don't see a reason that the parity there, even with R, is going to be large at all. So yeah, thanks for your time.
Cool. My mind's melting. Does anyone have questions whose brain isn't exploded from science? In the back. Okay, hold on.
One question. Is what you're working on right now, do you have to tabulate data from other medical studies, like from Harvard, for comorbidity on some of those inheritable diseases you were talking about?
A lot of those genetic studies are actually from multiple groups, and since you're not allowed to share a lot of the data is actually protected, so you're not allowed to, even between scientists, you're not allowed to share that data in dbGaP. And so when you want to add, actually a lot of the analysis that's done is basically a meta-analysis, where you try to combine the p-values together, which you lose statistical power in, but it's right now it's the only step to combine large studies. But there's actually, there's a lot of IT work in trying to get that so that doesn't have to happen. To protect the data, basically on encrypted data, to do an analysis on the encrypted data. But I don't know how far that is actually. It's not my realm of expertise, but that's an interesting point. The genetic data also, I don't know if this answers your question, but the genetic data also has other covariates, like let's say smoking. You include that in the regression analysis as a covariate. And so that will account for that. But again, in the meta-analysis, when you're combining multiple studies, like we combined our data with a group from Brigham and Women's Hospital, and so you combining the covariates actually quite hard, because you have to basically use the same covariates between the studies and then combine the p-values afterwards. So yeah, it's not as straightforward, but people are working on ways to combine the data in the protected data sets